Welcome mga kabayo! We will tackle the central dogma of molecular biology in our video series. At the end of this video series, we will be able to illustrate the molecular structure of DNA, RNA, and proteins, diagram the steps in DNA replication and protein synthesis, discuss the significance of the central dogma of molecular biology. Genetics plays a big role in our society. By having an in-depth understanding of genetics, we can really learn a lot of things that will help us survive. The central dogma of molecular biology is the basic underlying principle in the field of genetics. This explains that DNA codes for RNA, which codes for proteins. But before delving into this principle, we should have a deep understanding of the different molecules and structures of the following components: DNA, RNA, and proteins. Let us get started. DNA is a permanent storage place for genetic information. It controls the synthesis of RNA. The sequence of nitrogenous bases in the DNA determines the protein development in new cells. DNA is double helix to ensure that no disorder occurs. DNA runs anti-parallel. One strand of DNA holds the information that codes for various genes. This strand is often called the template strand or antisense strand. The other and complementary strand is called the coding strand or sense strand. DNA has three chemical components: phosphate, a sugar called deoxyribose, and four nitrogenous bases: adenine, guanine, cytosine, and thymine. The phosphate group in the DNA is composed of a phosphorus atom surrounded by four oxygen atoms. By itself, the nucleotide might have three phosphates. However, when joined to the growing strand of DNA, two of its phosphates are lost, and the remaining one attaches to another nucleotide's sugar. The sugar group in DNA is called deoxyribose. The 5-carbon structure of ribose in the DNA molecule is numbered based on its carbon atom. The carbon atom on the right side is assigned as number 1, and the numbering sequence runs clockwise. The last carbon in the ribose sugar is numbered as 5'. The small mark in the numbering distinguishes it from any of the numbers given to atoms in the other rings. Its importance will be discussed in detail as we learn about DNA replication and translation. The nitrogenous bases in the nucleotide can be classified according to the number of rings in their structure. Purines, which are adenine and guanine, have a double-ring structure, while the pyrimidines, which are cytosine, thymine, and uracil, contain only one ring in their structure. The uracil is usually found in RNA and serves as a counterpart of thymine in DNA. The nitrogenous bases undergo complementary base pairing, wherein each pair should contain a purine and pyrimidine. Each nucleotide is paired together by forming hydrogen bonds. In DNA, adenine is paired with thymine, while guanine is paired with cytosine. Uracil replaces thymine in RNA. RNA is a single-stranded molecule that is also composed of nucleotides with a few modifications. The sugar backbone of RNA is ribose. Also, as mentioned earlier, uracil replaces thymine as one of the nitrogenous base in RNA. When the cell needs to get information that codes for a specific protein, RNA will copy that information, which is stored in DNA. This helps the cell get the instructions needed to produce the protein, while keeping the DNA information intact. How DNA and RNA works will be discussed in detail as we tackle protein synthesis. Proteins are the final products in the central dogma of molecular biology. Proteins are composed of polymers of numerous amino acids known as polypeptides. The three-dimensional structure of a protein not only defines its size and shape, but also its function. The folding in a protein structure allows for interactions between amino acids that are distant to each other. There is a system by which the particular order of nitrogenous bases in DNA and RNA can be translated into specific amino acids in a polypeptide. The language of instruction in the mRNA is called the genetic code. As mentioned earlier, DNA codes for proteins. This process involves transcription and translation. DNA transcription is the first stage wherein important information in the DNA strand is copied into the mRNA. The next stage is translation, wherein the information sent by the mRNA is analyzed with the help of ribosomes. The ribosomes translate the mRNA code into the proper protein format. Make sure to watch our following videos as we discuss central dogma of the molecular biology.